Welcome to the PubMed Daily Review for April 28, 2026. Today we scanned 713 new open access papers. Here are the top 5 selected for detailed analysis. Trend 1 of 5, Cognitive Event Segmentation and Health. This trend links how people process experiences to various health outcomes and conditions. This cluster covers 209 papers. Representative work includes development of event segmentation in language and cognition, evidence from dwell times and eye movements, integrating the mystical experience questionnaire into a broader psychometric framework, English validation of the psychedelic experience scale and comparison of psilocybin and LSD sessions across 2 controlled settings, and association between obstructive sleep apnea and periodontitis, a systematic umbrella review. Trend 2 of 5, Immunity, Memory, and Materials. This trend explores how biological systems and materials interact to enhance defense, recall, and repair. This cluster covers 128 papers. Representative work includes nasal humoral immunity, the first line of protection against respiratory pathogens, living rent-free in your head, resident memory T cells, and flexural strength, monomer release, and wear of occlusal splint materials fabricated through conventional, milled, or 3D printed methods. Trend 3 of 5, Oral Health and Disease Markers. These papers explore the interplay between oral health conditions and systemic disease markers, highlighting diagnostic and therapeutic connections. This cluster covers 200 papers. Representative work includes a clinical algorithm for full mouth rehabilitation in patients with generalized severe tooth, where a case report, prevalence of peri-implant diseases in patients with type 2 diabetes mellitus, a cross-sectional study, and prevalence of osteoarthritis using cone beam in patients at the University of Cuenca. Trend 4 of 5, Genetics, RNA, and Development. These papers explore gene regulation, RNA function, and developmental processes across diverse organisms. This cluster covers 83 papers. Representative work includes Cantharellus guyanensis, Basidiomycota, Hydnaceae, a gourmet mushroom in the Brazilian Cerrado, identifying target genes for engineered genetic incompatibility in fish, and whole transcriptome insights into light-responsive non-coding RNA networks, regulating circadian clock and DNA repair in zebrafish. Trend 5 of 5, Cancer Omics and Microenvironment. This trend explores how molecular profiles and the tumor environment influence cancer development and treatment. This cluster covers 93 papers. Representative work includes serum proteomic analysis using gel-based liquid chromatography, tandem mass spectrometry, reveals differences between canine oral malignancies and non-malignant conditions, simultaneous representation learning of multi-omics and clinical outcome data via a supervised knowledge-guided Bayesian factor model, and KDM3B predicts expression signature, prognosis value, and immune characteristics of cancers, a pan-cancer analysis. Up next, our first paper out of 5. Single-cell RNA sequencing reveals pancreatic cancer microenvironment heterogeneity for precision therapy by Yaya Bai and Duo Zhou from Department of Gastroenterology, Ruijin Hospital, Shanghai Jiao Tong University School of Medicine, Shanghai, China. Let's break down its key contributions. Pancreatic cancer remains one of the deadliest malignancies, with survival rates stubbornly low. A major hurdle is the incredible diversity within both the cancer cells themselves and their surrounding microenvironment. Understanding this complexity is key to developing more effective, personalized treatments. This review explores how single-cell RNA sequencing is revolutionizing pancreatic cancer research. By analyzing gene expression at the individual cell level, scientists can uncover the intricate details of the tumor microenvironment, identify distinct cell populations, and understand their roles in disease progression and treatment response. Studies using scRNA-seq have uncovered critical insights. For instance, specific cancer-associated fibroblast phenotypes were found to support tumor growth. A chemo-resistant niche, involving tumor cells, macrophages, and exhausted T cells, was identified and linked to a key immune checkpoint molecule. Furthermore, distinct subtypes of pancreatic stellate cells were characterized, contributing to a fibrotic and immunosuppressive environment that hinders anti-tumor immunity. In conclusion, single-cell RNA sequencing is a powerful tool that is fundamentally changing how we study pancreatic cancer. By dissecting the tumor microenvironment in unprecedented resolution, we are uncovering new vulnerabilities. Combining scRNA-seq with other advanced technologies promises to pave the way for truly precision therapies, offering hope for better outcomes in this challenging disease. Up next, our second paper out of 5. B2M drives PD-1 inhibitor resistance in DLBCL through independent dual immune escape mechanisms by Ying Liu and Qingyuan Shun from Department of Oncology, 3201 Hospital, Hanzhong, 723000, China. Let's break down its key contributions. Immune checkpoint inhibitors, like those targeting PD-1, have become a vital tool in treating diffuse large B-cell lymphoma. However, a significant number of patients don't respond to these therapies. We need to understand why this resistance happens to develop better treatments. This study focuses on a key molecule, beta-2 microglobulin, or B2M, and its potential role in this resistance. To investigate this, researchers used DLBCL cell lines where they either knocked out or restored B2M. They then examined various molecular and cellular responses using techniques like immunohistochemistry, PCR, Western blots, and flow cytometry. They also performed co-culture experiments with immune cells and tested the effects of inhibiting MHCI or stimulating with IFN-gamma. This allowed them to pinpoint the specific mechanisms by which B2M impacts immune evasion. The results were striking. Knocking out B2M in DLBCL cells led to a significant drop in both PD-L1 and MHCI expression. This impaired the cell's ability to present antigens and activate T cells, as seen by reduced T cell activation markers and lower cytotoxic activity. Importantly, the ability of IFN-gamma to boost PD-L1 was completely abolished in B2M deficient cells, highlighting a critical link. These findings clearly demonstrate that B2M loss creates a dual immune escape mechanism. So, what does this mean for patients? This study reveals that B2M mutations are a key driver of resistance to PD-1 inhibitors in DLBCL, acting through two distinct immune escape pathways. It suggests that simply targeting PD-L1 might not be enough for these patients. Restoring the function of MHCI appears to be a critical step. This opens up exciting possibilities for combination therapies that simultaneously address both MHCI presentation and PD-L1 signaling, potentially offering a new way to treat patients who currently have limited options. Up next, our third paper out of 5, Preclinical Nanoparticle Approaches Targeting Tumor-Associated Macrophages in Breast Cancer. From Mechanisms to Therapeutic Strategies by Jiahui Teng and Liangliang Wang from the First Clinical Medical School, Zhejiang University, Zhejiang, Jiangxi, People's Republic of China. Let's break down its key contributions. Breast cancer continues to be a significant global health challenge, particularly for women. While immunotherapy has shown promise, its effectiveness is often hampered by the complex tumor microenvironment. A major component of this environment is the tumor-associated macrophage, or TAM. These cells can unfortunately promote tumor growth and help cancers hide from the immune system. This review explores how nanotechnology can be used to target these TAMs, potentially unlocking new treatment strategies. This review systematically analyzed existing research on nanoparticle-based strategies for targeting TAMs in breast cancer. The focus was on preclinical studies, clinical trials, and mechanistic investigations. The researchers examined various nanoparticle approaches, including those designed to reprogram TAMs, deplete them, or inhibit their recruitment. The goal was to understand how these strategies impact TAM function and ultimately influence treatment outcomes. The review highlighted that TAMs play a dual role, but predominantly promote tumor progression through angiogenesis, immune evasion, and metastasis, especially the M2 subtype. Conversely, M1 TAMs exhibit anti-tumor properties. Several nanoparticle-based strategies were explored, including reprogramming M2 TAMs to M1, depleting TAMs, and inhibiting their recruitment. These approaches aim to shift the balance towards an anti-tumor immune response. In conclusion, targeting TAMs with nanoparticles represents a significant opportunity to improve breast cancer treatment, particularly in overcoming resistance to current immunotherapies. However, challenges remain in translating these findings from preclinical models to clinical practice. Future efforts should prioritize the development of human-relevant models, optimize manufacturing, and explore personalized therapeutic combinations to maximize patient benefit. Up next, our fourth paper out of 5, Single-cell and spatial transcriptomics reveal that the CXCL12 CXCR4 axis drives the immune desert phenotype in small cell lung cancer by recruiting immunosuppressive CXCR4 plus neutrophils and S100A8 plus monocytes by Peng Zheng and Rong Shun from Department of Oncology, Southeast University, Zhongda Hospital, Nanjing, Jiangsu, China. Let's break down its key contributions. Small cell lung cancer, or SCLC, is a particularly aggressive form of lung cancer. Unfortunately, it doesn't respond well to current immunotherapies, largely because its tumor microenvironment is highly immunosuppressive, creating what we call an immune desert. We don't fully understand the molecular reasons for this, which is a major hurdle in finding better treatments. To investigate this, researchers used a combination of cutting-edge techniques. They analyzed both small cell lung cancer and non-small cell lung cancer samples using single-cell RNA sequencing and spatial transcriptomics. They also employed multiplex immunofluorescence to validate findings and used animal models to test their hypotheses. This allowed them to pinpoint specific cell types and molecular interactions driving the immunosuppression. The study revealed significant differences between SCLC and NSCLC. SCLC tumors had more than double the proportion of neutrophils, and these neutrophils, along with monocytes, adopted an immunosuppressive profile. Crucially, the CXCL12 CXCR4 signaling pathway was identified as a key driver, recruiting the suppressive cells and hindering T-cell activity. When this pathway was blocked in animal models, tumor growth decreased and anti-tumor T-cells increased. In conclusion, this research strongly implicates the CXCL12 CXCR4 axis in creating the immunosuppressive environment of SCLC. This finding is clinically significant because it suggests that targeting this axis could be a promising strategy to make SCLC tumors more responsive to immunotherapy. Further studies are warranted to explore the therapeutic potential of CXCR4 inhibitors in this challenging cancer. Up next, our fifth paper out of 5. Targeting STAT5 via CRISPR/Cas9 restores TKI sensitivity in resistant chronic myeloid leukemia cells by Besencelik and Bursan Teskamli from Medical Biology, Faculty of Medicine, Edge University, Izmir, Turkey. Let's break down its key contributions. Chronic myeloid leukemia, or CML, is often treated with tyrosine kinase inhibitors, or TKIs. While effective, many patients develop resistance, making treatment difficult. A protein called STAT5 has been identified as a key player in this resistance. This research asks if targeting STAT5 could be a way to restore the effectiveness of TKIs in CML. To investigate this, the researchers used a powerful gene editing tool called CRISPR/Cas9 to knock out the STAT5A gene in both standard and TKI-resistant CML cell lines. They then assessed various cellular responses, including cell viability, apoptosis, and cell cycle progression. They also analyzed gene expression changes and used computational tools to understand the underlying molecular mechanisms. This multifaceted approach allowed them to thoroughly examine the role of STAT5A. The results were quite striking. Knocking out STAT5A led to a significant decrease in cancer cell viability and a notable increase in apoptosis, meaning the cells started to self-destruct. The cells also showed arrest in the G0/G1 phase of the cell cycle, halting their proliferation. Furthermore, the expression of key genes involved in the JK/STAT pathway and apoptosis was altered, indicating a reprogramming of cellular signaling. In conclusion, this study strongly suggests that STAT5A is a critical target for overcoming TKI resistance in CML. The ability of STAT5A knockout to restore sensitivity highlights its potential as a therapeutic strategy, possibly in combination with existing TKIs or STAT5 inhibitors. While these findings are promising, further research in more complex models and patient-derived samples is needed to fully explore its clinical applicability. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.